We are conducting an experiment to observe the phenotypic ratios in fruit flies, specifically the ratio of red eyes to white-eyed flies, with red eyes being the dominant trait. Over the course of 5 weeks, we will count the number of red-eyed flies to white-eyed flies before breeding them and releasing the adults so that only the eggs are left. The phenotypic ratio will then be recorded each week, and then we will compare the ratios after the experiment is completed.
So for our hypothesis, we hypothesize that when the fruit fly phenotypes are observed through each generation, the red-eye phenotypes will occur more frequently than the white-eyed phenotypes.
And then for our predicted outcomes, we said that throughout the course of the 5 generations, we will see an increase in the red-eyed phenotype. And we will also, for each generation, we are also going to see a higher ratio of red-eyed individuals compared to the white-eyed individuals, as well as at least one generation throughout this whole experiment will be fully red-eyed, and we will have to add variation into it.
Some background on these organisms. In our organisms, we have red-eyed and white-eyed phenotypes. Red eyes are considered the wild-type phenotype, while white eyes are a recessive trait. In each generation, we have both male and female flies of both eye types. The females are larger and have lighter, more pointed abdomens than males, who are smaller with darker, rounded abdomens.
HWE violations. The HWE assumptions we are violating is that the population is facing outside change, since we will be adding variation if only one phenotype is present in the tube. We will add the missing phenotype. We will also be interfering with the population if we end up having all males or all females in the tube, which would then lead us to add the missing sex.